We're all here to honor the work and evaluate the work by Justin Shaffer. Thank you for coming in particular between a holiday weekend and a holiday. I think that officially makes today a holiday, so we're really glad that you're here. Just a few introductory thoughts, and then I'll turn things over to Justin's co-advisor, Dave Walters. Dave and I have had the honor of working with Justin since 2012 when he came to the University of Arizona and joined the plant pathology program. Over the ensuing years, I've really benefited from working with Justin, as I would say all of the members of our lab have. Justin has been a wonderful contributor. You guys can come on in, there's these filter in. There are four things that I just want to highlight about Justin and thank him for the experience that we've had together. The first is just the degree to which Justin has developed as a mentor and an ambassador for science. As many of you know, in this day and age, outreach for science is perhaps more important than ever. And Justin has done a tremendous job, especially in reaching out to undergraduates and high school students. And I think that if we were to add the number of people with whom Justin has interacted about science over the last 5 years, it would be easily 800 to 1,000 when we consider high school students, members of the public, and many different roles that Justin has played. So I'm really proud of that, and that really means a lot to me. More locally, Justin also has been a tremendous mentor of undergraduates. He's worked with 8 undergraduate students, including an opportunity to mentor students all the way to presenting at national meetings, which is really a tremendous achievement. The second thing that I'd like to highlight about Justin is his incredible activity as a teacher. One thing that I've had the opportunity to do is to sit in on his section when he's been teaching laboratory courses. And in interviewing students in the room, I asked them if they could describe their experience. And 201, they didn't know I was coming. Each one said, "Justin is awesome," which I think is about like the best teaching validation we could ever hope for. I don't think I've ever gotten that. The third thing out of 4 is that I just want to highlight Justin's achievements. So he has been incredibly successable in terms of many of the markers of achievement in science and in publishing, and certainly getting 2 papers out already from his dissertation. Those are both highly cited already and attracting a lot of attention. Also, he's received support from the Mycological Society of America in the form of competitive awards. He's also been supported by the School of Plant Sciences, by a fellowship award through plant pathology and so on. So Justin is very accomplished in all of those ways. And then finally, last but not least, I just want to compliment him on his research. So Justin took on a challenging project, challenging in terms of concept, technical aspects, computational aspects, and and he was successful in achieving what I think is a very interesting and integrative body of work as a graduate student. So I'm really happy that we're all here today, and I want to thank you guys for coming. And those are my thoughts as one of Justin's advisors, and let me turn over to Dave as number 2.
We'll keep this short and sweet. I'm just echoing everything Betsy said. Justin has been a tremendous TA, a tremendous part of my lab, and interacting with the people and undergraduates that I've dealt with over my years here. And just to finish up on a very positive note, Justin, I think his application was in the first batch of applicants that I saw when I first started here in 2011. I was on the graduate committee. His application came through, looked tremendous. And then we lost him to Oregon State University, which for me personally hurts a little. I'm an Oregon Duck through and through. But then a year after that, we got him back. So a positive note, he decided to join us, and has been a tremendous addition. He's my first Ph.D. student that's actually going up, and just the intellectual curiosity and the drive and kind of just the conversations we've had over the years in science have been great. So that's Justin Shaffer.
Thanks, Dave and Betsy. It's been an honor to work with both of you, and you're both awesome. Thanks for coming, everyone. I'm really excited to talk about my research. For those of you in the back that maybe can't hear me, just raise your hand and I'll try and project better. And for these rooms with 2 screens, I'll be using my pointer throughout, and I'll be using it on this right screen, if you're interested. Okay. I would like to first start with this panel of images showcasing plant-fungus interactions. Fungi are particularly interesting and also functionally diverse with respect to their interactions with plants. So as far as interacting with plants, fungi can alter photosynthetic processes. Fungi can also alter nutrient and water uptake, affecting things with roots, and also stems, which also influences structural integrity of plants. Fungi that use flowers to grow and reproduce, affect reproduction of plants, and even fungi that are living within floral parts, such as the nectar, can influence interactions with pollinators. So I'm really interested in the factors that influence the stability of plant-fungus symbioses through both short and long time scales. And so a lot of work has shown on both the fungal and plant host side that genetic factors are really important. So what plant you are and what fungus you are is really important. But also where these interactions are occurring in the environment. So things like temperature and moisture we know are really important. In general, things that influence the fitness of the fungal partner or the plant host are what we think about as being important factors. More recently, there's been growing evidence that the occurrence of additional microbes in these systems is really important for the outcomes of plant-fungus interactions. And one really important group of microbes that associate with fungi are bacteria. And so what I'm showing you here on the left are some micrographs of bacteria tagged in green within fungal spores. These bacteria are also tagged green within the spores of this fungus. And these bacteria are tagged yellow, living within the vegetative cells or the hyphae of this fungus. These bacteria are called endohyphal bacteria because they live within fungal cells. And I just wanted to walk you through a couple of really well-studied examples of bacteria and fungi that can modify the phenotypes of their hosts. A really well-studied example is that of Rhizopus microsporus, which is a rice pathogen. This fungus harbors bacteria closely related to known Burkholderia, and these bacteria are responsible for the virulence and reproduction of this fungus. When you remove the bacteria from this fungus, it is no longer pathogenic on rice, and it also loses the ability to produce sporangia. Another really well-studied example are bacteria that live within our muscular mycorrhizae within the family Gygosporiaceae. Of which a member is listed here, Gygospora margarita. And bacteria in these fungi influence pre-symbiotic phenotypes, so how the fungi behave before they find our host plants. So things like growth, detection of host plant roots are influenced by these bacteria. The final example is an ascomycete fungus called Pestilodiopsis neglecta, which harbors a bacterium related to known Lutobacter that can influence auxin production by the fungus. This last example is particularly interesting because it's a member of the Ascomycota, which is a distinct group relative to the fungi listed here, both morphologically in that the fungal cells have cross walls that can limit the movement of things within the cytoplasm, unlike these fungi, which are what's called cenocytic and are just continuous. So you can imagine for bacteria, this might be an important factor. Additionally, the Ascomycota is the most species-rich group of fungi, and they're really well known for their interactions with plants. This group of fungi diverged roughly 500 million years ago. We have described just shy of 100,000 species, but we estimate anywhere from 400,000 to 7 million exist. This group has diversified with plants for over 700 million years, and there are multiple origins of symbioses within this group, so they do many different things with plants. Some are pathogens, some interact with plants mutualistically, and so on. A lot of the diversity in this group are associated with tropical plant species, and in addition to influencing individual plant health, in the tropics, fungi have been associated with plant population as well as community dynamics. As an example, fungi that associate with seeds are particularly interesting in these forests because seeds are the most common form of reproduction for most plants. I just want to emphasize that when I'm talking about fungi associated with seeds, I'm referring to those fungi that recruit to seeds after their dispersal to the forest understory. So these are not fungi that are vertically transmitted through seeds from mom to offspring. They're likely originating in the soil. And work has shown that there's great taxonomic and functional diversity in these fungi. So here I'm showing some of the diversity of these fungi growing on seeds here. And work has also shown that these fungi are closely related to fungi that are growing in co-occurring leaves. So these pictures are showcasing emergent fungi coming out of leaf pieces here. Seed-associated fungi are also generalists in that they can colonize the seeds of many different plant species. And they have significant effects on the outcomes of interactions with seeds, such as seed germination and survival. So my overarching research question is, how do endohyphal bacteria influence seed-fungus interactions? And I'd like to outline a couple of my research aims just to orient you. My first aim is to describe the occurrence, diversity, and evolutionary relationships of bacteria among these fungi. I'm going to focus on two particularly important groups, the Xylariales and the Hypocoreales. These are very species-rich groups of plant-associated fungi. The Xylariales is comprised mostly of saprotrophic or endophytic species that move from leaves to the forest understory. And the Hypocoreales are well known for soil-borne plant pathogens. I'm then going to explore whether or not these bacteria have effects on their host fungal phenotypes, and then carry that a bit further to explore whether or not those effects would carry to seed fungus interactions. And we're going to do this through some seed germination assays. I'm going to end by presenting some ongoing work and future directions involving the development of some methods for evaluating gene expression in these fungus-bacterium pairs involving Ascomycota. Great. To begin with associated fungi. And in general, we found that these bacteria are common and diverse among these fungi. Again, I focused on two different groups. And first, I'm going to bring up the species that were represented among the fungi screen. And then the species of endohyphal bacteria that were detected among those fungi. And so you can see here, and by the way, these are non-singleton bacterial species, so this doesn't include singletons. We found 27 species of endohyphal bacteria among 6 species of Xylariales, and 38 species of endohyphal bacteria among 12 species of Hypocoreales. The rough occurrence of these bacteria is around 80%, which is shocking for bacteria among Ascomycota. It's higher than any rate that we've seen. I would like to summarize the phylogenetic placement of these bacteria among these two groups of fungi using this phylogeny of bacteria here that shows the major bacterial phyla in addition to some classes of proteobacteria. When we look at the distribution of endohyphal bacteria among the Xylariales, we see that the majority of bacteria belong to proteobacteria, but we also have some gram-positive groups, such as actinobacteria and firmicutes. The distribution of bacteria among the Hypocorealian fungi is very similar, although we see members belonging to 2 additional phyla popping up. With respect to endohyphal bacteria that have been discovered in non-Ascomycota, so the fungi that I talked about earlier, the endohyphal bacteria that we detect among our Ascomycota are phylogenetically distinct. And I'd like to showcase that by showing you a trim phylogeny of bacteria in the Burkholderia ACE. This phylogeny is based, again, on the 16S ribosomal RNA gene, and it is a best tree from a maximum likelihood analysis with support from 1,000 bootstrap replicates and Bayesian posterior probabilities. I'd like to first point out in blue endohyphal bacteria that I discovered among seed and leaf-associated fungi in this work. In gray, I've highlighted Burkholderia and relatives discovered previously in non-Ascomycota, the ones that I discussed earlier. The majority of endohyphal bacteria in non-Ascomycota belong to the Burkholderia ACE, and it's interesting that the bacteria that we're finding are distinct from those. The phylogeny that I'm going to show you next is a fungal phylogeny. It's Entenema, which is a member of the Xylariales, and this is a very similar tree, but it's based on fungal ITS and partial LSU ribosomal DNA. What I wanted to show you is that we found indeed that seed-associated and foliar endophytic fungi often have a shared evolutionary history Here, I've highlighted all isolates included in this analysis that were derived from leaves in green, all those derived from seeds in brown. If I focus the highlighting to include only those isolates that I screened in this study for the presence of endohyphal bacteria, and then bring up the results of that screen where pluses indicate, yes, this fungus had bacteria, and where white circles indicate the presence of a unique bacterial species, you can see that all of these fungi had bacteria in them, and they all had a distinct bacterial species. Some cases even have 2 distinct bacterial species present. And while these fungi, seed and leaf-associated fungi, have a shared evolutionary history, we found that they harbor distinct endohyphal bacterial communities. And so the way we explored that was using non-metric multidimensional scaling, which I'm showing a plot of here, where each point represents a fungus that's separated from all other fungi based on the similarities in their bacterial communities. And so points that are really close to each other have very similar bacterial communities. Points that are very distant from each other have very distinct bacterial communities. And while you can see that there is some overlap in the bacterial communities of seed-associated fungi in brown and foliar-epiphytic fungi in green, there are also fungi that harbor distinct endohyphal bacteria. This is supported by 2 independent statistical tests. And so just to summarize from this initial aim, we found that endohyphal bacteria were common, phylogenetically diverse, and distinct from bacteria in non-acid mycota. As far as the fungi, these 2 groups have a shared evolutionary history. However, they have distinct endohyphal bacterial communities, which may speak to the ecological modes of the fungi with respect to some living in leaves versus some living in seeds. So because of previous work showing the importance of endohyphal bacteria for fungal phenotypes and how they interact with plants, the next step is to explore whether or not these bacteria influence our fungi. To do this, we utilize a phenotypic microarray where we're comparing substrate use between fungi that have endohyphal bacteria, what I'm going to call EHB plus strains, and fungi that have been cured of their endohyphal bacteria using antibiotics that I'll refer to as EHB minus strains. And I've also denoted that here that we derived this fungus over here that does not have bacteria in it from the naturally infected strain that we derived from seeds. To carry out this assay, we used a biolog microplate that was designed for filamentous fungi. This microplate has 95 unique carbon sources in it. It allows for measurement using 2 independent ways. One of them is through cellular respiration, and so we'll see a color change if the substrate is being used. And the other through it is a proxy for growth, just hyphal density. In order to focus in on one fungus-bacterium pair, we decided to select a member of the Fusarium solani species complex, which is a group of very well-known plant pathogens that has a very wide host range. I'm showing you here a diseased potato that's been infected with Fusarium solani, as well as a giant field of strawberries that has been attacked by Fusarium solani as well. We selected specifically a subspecies called Keratoplasticum. This specific isolate harbors a bacterium related to those in the group Chytnophaga, and this group, some members of this group are known to have chitinolytic activity, which is important for interactions with fungi because fungal cell walls are largely made of chitin. So once we obtain pure cultures of our cured EHB minus and our EHB plus fungal strains, we create hyphal suspensions that we then use to inoculate the microplates, and we then measure every 6 hours for about a week. After 1 week, the microplates become fully grown and the experiment is over. We repeated this experiment 2 times with mirrored results, and I'll be presenting results from the first run. This is what one of the plates looks like at the end of the 7 days. Again, the color change that you're seeing here corresponds to substrate use based on cellular respiration. And so I just wanted to walk you through some of the major groups of compounds that are in these plates so that you can see how they're being used by this fungus. First, we have simple sugars, which you can see are kind of pink, so they're being used at a moderate level. We also have amino acids, which are used quite a lot, as well as carboxylic acid, which have variable use depending on which substrate there is. As far as our expectations of substrate use, water is kind of the control on these plates. So for something like water, we would expect minimal growth, and we see that here. For some sugars, such as mannose, we would expect some growth here, indicated by the pink color. And then for something like an essential amino acid, we would expect complete use of that substrate. Great. And so now I'm going to use one example to show you that in some cases, the EHB plus strain outperformed the EHB minus strain. Okay. Here I'm showing you glucose as an example. On the y-axis, we have hyphal growth, which was highly correlated with measurements for cellular respiration. So for the remainder of this part, I'll be showing you data for hyphal growth. And on the x-axis, we have time. And what should pop out to you is that after roughly 2 days of running the experiment, the EHB plus strain, the fungus that has endohyphal bacteria, significantly outperformed the EHB minus strain. When we looked at substrate use across all substrates, we found that the greatest difference was at 7 days, the end of the experiment. And so we used that point, 7 days, to analyze differences in global substrate use. Substrate use across all 95 substrates between fungi that EHB plus and EHB minus fungal strains. What I'm showing you here is a cluster dendrogram where each tip represents one phenotypic microarray, so one biolog plate. They're being clustered to all other plates based on similarities in substrate use across all substrates, so global substrate use. And what you can see is that plates inoculated with EHB plus strains are utilizing substrates more similarly to one another than the EHB minus strains are. Okay? And that's indicated here by all of the EHB plus plates grouping together before they start clustering with any of the EHB minus plates. This was supported by a permutational multivariate analysis of variance. And in general, we saw, while there was differences in use across all these substrates, we saw 4 major patterns in substrate use between EHB plus and EHB minus strains. The first pattern was negligible growth by both strains. So neither strain was able to grow on these substrates. This included things like alpha-cyclodextrin, which is a synthetic compound which we may not expect these microbes to grow on because they've never seen it before. But we also see some things like alanilamide, which is an antibiotic. The second major pattern we saw was that they both grew, but they didn't have a difference in growth. And we saw this across 13 substrates, including mannitol, which is highly abundant in plant tissues, as well as aspartic acid, which is important in seeds. The third pattern we saw was that the EHB minus strain outperformed the EHB plus strain. And this was only for a select group of substrates, including rhabinose. It's a form that's not common in nature, as well as succinamic acid, which is a precursor to many plant growth-promoting compounds. However, the most common pattern is that the EHB plus strain outperformed the EHB minus strain. We saw this across 60 substrates. I want to highlight some of these substrates with respect to their importance in plants and seeds. So some important substrates we saw are important constituents of seed coats. So these are things that would be important for a fungus colonizing a seed, for example. We also see things that are mobilized or produced during seed germination, which is a very important process. And we also see some important global regulators. They're endohyphal bacterial partners. And again, I'll remind you that some of these fungi harbor multiple endohyphal bacteria. So in addition to representing fungi isolated from both seeds and leaves, I'll remind you that these are hypocorallian and xylarian fungi, 2 of the most important groups of plant-associated fungi. However, because of time, I'm going to focus and discuss results from only fungi that we got from seeds. Okay? Just some brief methods. Again, I'll remind you that we're comparing EHB plus and EHB minus fungal strains. Once we have those fungi in pure culture, we go out into the forest and collect seeds either right off the tree or from fruits that were recently fallen beneath the tree. We wash all the seeds and surface sterilize them to remove any surface microbes that might interfere with our experiments. We then plate them on petri dishes that are completely grown with fungi, alongside controls where seeds are just placed on agar plates. We used 5 plant species in this experiment. We have 6 fungi, and for each plate, we had 20 seeds and 5 replicates for each seed fungus combination for a total of 7,000 seeds in the experiment, including controls. We allow the seeds to be colonized for roughly 3 weeks, which we determined to be sufficient in preliminary experiments. After which we transfer seeds to moistened filter paper that we then place in the shade house where we measure germination weekly for about 7 weeks. The 7-week cutoff, the cutoff for germination studies is typically the point at which those seeds have germinated for 2 weeks. Just some very interesting results. We saw very different abilities and differences in the abilities of fungi to colonize these seeds. Here I'm showing you 4 classes that we recognize, going from colonization that you need to look at with a microscope to colonization that you can't see the seed anymore under. However, interestingly, while we saw differences in the abilities of fungi themselves to colonize seeds, we did not see a difference in the ability of strains, so DHB plus and DHB minus strains of the same isolate to colonize seeds. And so what I'm showing you here are some colonization data where I have the seed colonization on the y-axis and bars representing the number of seeds colonized by the DHB minus strain and the DHB plus strain of this particular fungus on the x-axis. And what you can see here is that both strains have similar colonization rates. This was such for other fungi on seeds of the same species, and in general, this was the pattern. However, in some cases, we did see subtle variation, which the ecological importance is not known. While we didn't see differences in colonization between DHB plus and DHB minus strains, the presence of bacteria did influence germination of seeds. So here, in a similar framework, I'm showing you the proportion of seeds that germinated on the y-axis by the endohyphal bacteria cured, the strain that does not have endohyphal bacteria, the DHB minus, and the DHB plus strain of this fungus. The gray lines indicate the mean germination rate for control seeds, so seeds that did not that were not exposed to fungi. And the bars highlight deviations from that control germination rate. And what's very interesting is that while strains that were cured of their endohyphal bacteria have significantly reduced germination compared to uninoculated control seeds, the DHB plus strains, seeds infected with the DHB plus strains, I should say, germinated much more similarly to uninoculated controls. This pattern was true for other fungi on the same seed species. And in general, we saw this case where endohyphal bacteria seem to reduce the negative impacts of these fungi on seeds. However, based on the fungus, the bacterium, and the seed species that we put these microbes on, the outcomes can often be context dependent. So oftentimes we see the opposite case. I'll just last minute point out that these asterisks indicate significant deviations from this control, and the brackets indicate significant differences between DHB plus and DHB minus strains. Great, so just to summarize that component, we found that these bacteria significantly influence these interactions, including germination. In general, the negative impacts of fungi are reduced by the presence of these bacteria. But again, the outcomes are context dependent on the players at hand. So I would like to I would like to finish with discussing some ongoing work and future directions. We are developing some methods for evaluating gene expression in these Ascomycota. And I would just like to give you guys some background on how gene expression in these fungus bacteria pairs has been studied in the past. And so early on, you can imagine people focused on specific genes of interest. So for fungi, they're interacting with bacteria, that might be something like the ability of the fungus to detect its host plant. And so genes that were of interest there would be, for example, genes that help mycorrhizal fungi detect their host plant roots. On the bacterial side, people have focused on genes involved in interacting with eukaryotes, so things that are components of secretion systems, and other things for interacting with hosts. More recently, there has been transcriptomics work, so looking at gene expression across the whole genome. However, it's typically focused only on the fungal partner, ignoring gene expression differences in the bacterium. And there's two main reasons why I think this might be. One is that often the bacteria are convoluted symbionts, and you cannot grow them in pure culture. And this is the case for bacteria that are among the arbuscular mycorrhizal fungi that I mentioned. However, I suspect that it's our bias for these fungi because they are the partner that's interacting directly with the host plants, right? These guys are interacting while the bacteria is inside. And while studies like this have been very informative, I would argue that we need to look at both partners, gene expression changes in both partners to get a comprehensive understanding of these dynamics. One last note is that gene expression studies have not yet been conducted in Ascomycota that have endohyphal bacteria, which is particularly interesting and why we have decided to use the endophytic Ascomycete, Pestalozziopsis neglect, as our model for testing this culturing and gene expression pipeline. So I just want to tell you a little bit about this fungus. This fungus hosts endohyphal bacteria called Ludobacter, and this bacterium has been shown to influence both enzyme and hormone production. This work was conducted using the EHB-, EHB+ framework, and I'd like to show you some interesting results from that work. The first here is involving the production of auxins, so hormones that are important for plant growth. On the y-axis here, I'm showing you the concentration of auxin being produced and measured in the culture filtrate of a fungus that has its bacterium and the fungus that's been cured of its bacterium. What we can see is that over time, the EHB+ strain is producing more auxin than the EHB- strain. When these culture filtrates are applied to the roots of tomato seedlings, you see great differences in growth between seedlings that are inoculated with culture filtrate from the EHB+ strain versus the EHB- strain, just highlighting the importance of these interactions for plants. The bacteria can also be grown ascetically, which is great. And it is in part reliant on its host, even though it can be grown in pure culture. And I'll mention this in a bit. It has to do with sulfur acquisition from the medium. We have genomes available for both partners, and up to now, work on those genomes has shown that in addition to a lack of a reduced genome in the bacterium, so it has a normal genome size, the bacterium is lacking major sulfate transport genes, which is where this host dependency is coming from. And so that's one major factor here in this system. The bacterium is also lacking type 3 secretion machinery important for interacting with eukaryotic hosts such as fungi. Type 3 secretion systems have been indicated as being crucial for the symbiosis between the rhizopus pathogen and its Burkholderia symbiont. And so this is very interesting that it has this dependence on its host, but it doesn't have all the stuff that it would need to interact with hosts per se. So just to outline briefly our culture pipeline for looking at differential gene expression in these Ascomycota in general. The first step would be to optimize conditions for each microbe growing by itself. Okay? So in our case here with Pestalozziopsis and Ludobacter, because Ludobacter has this sulfate dependency, we're going to use a high sulfur medium. Okay? It's going to ramp up the bacterial growth to exponential phase such that we can then induce association with the fungal host. We do this by replacing the medium with a low methionine medium and make the bacterium seek out its host. Okay? We then create the culture from inoculum from the fungus and bacterial Bayesian cultures that we then replicate for the purposes of RNA-seq and then extract RNA for sequencing. And in this case, we're going to use Illumina technology. And although this work is ongoing and we hope to use this pipeline for other systems, I'd like to share with you some preliminary results that we have concerning the growth prior to RNA extraction in this Pestalozziopsis system. What was very interesting is that we found the fungus to grow to a higher biomass when it was grown alone compared to when it was grown with bacteria. And so here I'm showing on the y-axis the biomass of the cultures, and you can see that the fungal biomass is much higher by itself, even when we include the weight of the bacteria in a cold culture, which implies the fungus is much happier on its own. We see the opposite case in Ndobegang, which we might expect, given that it has some level of host dependency, where we see bacterial cell density now on the Y is much higher in the cold culture when the bacterium is with its host fungus, as opposed to when the bacterium is by itself. And so when we look at differences in gene expression, we expect these differences to reflect in part for things involved in sulfur dynamics there. But this new result with the antagonism of the fungus by the bacterium, which was not clear before this growth experiment. And so things we might look for along these lines are chitinase activity. Does this bacterium have the ability to eat this fungus? We might also look for the bacterium's ability to regulate secondary metabolites produced by the fungus, so kind of defenses, as well as production of secondary metabolites by either partner. Because of this antagonism of the bacterium on the fungus, you might expect the fungus to be responding in some defensive way, and we might see that the bacterium has some way of dealing with reactive oxygen species, things like that. Great. So just to summarize broadly, we found that these bacteria are common and phylogenetically diverse in these two groups of fungi. They can significantly influence not only fungal phenotypes, but seed fungus interactions, including germination. The outcomes of these tripartite symbioses are often context dependent on the partners that are at hand. But in general, I feel that the effects of these bacteria is really important for the ecology and evolution of these fungi. And more broadly, these interactions demonstrate not only the complexity, but the importance of these cryptic symbioses for things that we can see with our naked eyes, such as plants, but they're being influenced by these tiny microbes. So I'll end with the slide that I started with just one added thought that these interactions are still very interesting. Fungi are still influencing almost every function of plants, but now we know about this extra level of complexity. There are these bacteria there. And so I hope that future work will explore the importance of not only endohyphal bacteria, but bacterial fungal interactions in all of these emergent symbioses. And with that, I would like to thank a number of people, including my committee members, Rachel, Mark, Jeremiah, in addition to Betsy and Dave. I have had a really good time working with you guys. You guys have been great every time we meet. I feel very enlightened, and I come out of the room a better, smarter scientist and individual. I could not have asked for a better committee, even though I've only had one. I think this is, I don't know, you guys get along so well, and you seem like a very good team. I would really like to thank everyone in Betsy and Dave's groups. It's really hard to work on your own. It's really nice to have people around. And so, asking questions, help with methods, ideas. Specifically, I'd like to thank everyone that I came in with who may or may not be here now. So Kayla Arendt, Ryan Smith, Dustin Sandberg, and more nowadays, Joe Spraker, Kayla Garcia, Ming, Arnold Lab, and Paul Scott have been very helpful. I'd also like to thank many UA faculty, particularly those that I was instructed by. You guys definitely molded my trajectory, and I can't thank you guys enough for instilling in me the ideas that you have. There's also been a number of research staff members that have assisted not only my teaching, but also my getting paid and really important things like that. Actually, that would be the administrative staff. Hopefully the teacher staff is kind of included my getting paid. But especially Connie and Nancy, who are helping in all the labs that I've been instructor for. Gilberto has been very helpful in letting us borrow things and including ideas from his lab. Also Tim in the Mosier lab, Andrew Nelson in the Beilstein lab, Tim Rast, and Derek Swickle. There's a number of grad students, you guys all know who you are. You were very helpful and encouraging and motivating along the way. And I could not have done it without you guys. I wouldn't have done it without you guys. And so thanks. I'm not going to mention specific names. You guys know who you are. I'd like to thank my collaborators at the Smithsonian Tropical Research Institute. People there were very helpful. You can imagine doing science in, I'll use the word developing world, developing countries, not the best word, but you can imagine doing science in a place that's not the US is very difficult. And so having support in addition to motivation is very, very, very nice there. I'd like to thank funding sources, including the Smithsonian, but also the Mycological Society, School of Plant Sciences, and other funding sources on campus. I've been very lucky to obtain intramural funding from the U of A, and I'm very thankful. And I would just like to end with thanking friends and family, lots of friends and lots of family, and lots of help from both along the way, especially from my mom. Thanks, Mom. Really appreciate all your support this whole time. Great, and with that, I'll take any questions you guys might have. Thank you. Yes, very on.
That was a really great tool and fascinating work to communicate to someone who's not in your field. I really enjoyed it. So I have a question, and hopefully this will show my misunderstanding of it. So you showed that the bacteria reduce the negative impacts of the fungi on the seeds and showed that they increase seed germination when they have those bacterial relationships. So I was just wondering if you have any idea, maybe you didn't get into this yet, but like what are the bacteria doing, like why do they increase the germination rate? And that sort of seems to imply to me that they negatively impacted the fungi. And if that's true, that seems to sort of contradict some of the other things that show that the bacteria actually like enhancing the fungal performance.
Great. Great. So Mariel asked a very good question of what do you think is happening? How are the bacteria reducing the abilities of the fungi to hurt seeds? And this phenomenon where we see the fungus being antagonized by its bacterium seems to counter everything that I told you about these bacteria initially. So I think the bacteria may be parasites of the fungi, and so when the fungi do not have their bacteria, they are seed pathogens, and they are going to be detrimental to the seed. But perhaps the bacteria are either causing them to not be able to do that by being a parasite, or they may be providing resources for the fungus that then they don't need to attack the seed. And so they just grow on the seed as like an epiphyte. I think it could be either one. As far as the phenomenon of seeing this negative interaction for the first time, I would agree with you that it is quite surprising. Usually people talk about these interactions in the context of the bacteria helping the fungus do something or helping the plant interact with the fungus in a positive way. And I would just say that there's probably a diversity of interactions out there that we haven't found yet, and this is just the other side of the coin. Yeah. Any other questions? Yes? Dr. Pryor.
Oh, Justin. I appreciated the image that you showed at the beginning, the fluorescent staining and showing the live cells and the dead cells of the bacteria, interhyphal. Do you have any sense of interhyphal population of these bacteria? Because I'd be thinking that hyphae is going to be packed with bacteria. And do you have any information on numbers of some of these, or population of some of these associations? And if not, how would you go about getting those numbers?
Sure. Getting quantitative data along those lines has been difficult, pretty much because the distribution and abundance of the bacterial cells within the fungal hyphae is very heterogeneous. One thing that I can say is that in older parts of the tissue, you will see different dynamics than in newer parts of the complement of the fungal colony. Say that the
treatment, do you see differences in those categories? Like, for example,
yes. So Zhang is asking, I showed you 4 colonization classes, but then the graphs I showed you, they all have the same level of colonization. We did see for isolates, they would have number 4, or some isolates would have number 1. And within those isolates, both strains, DHB plus and DHB minus, would have that level. Does that answer your question?
so it's like, basically your colonization percentage can be translated to that percentage within each category.
I would just say that the factor of DHB is not important in that analysis. So you would get the same colonization, like the number of seeds colonized for either strain of a particular isolate. The isolate itself may have high or low colonization, but strains within the isolate will be the same.
So So would you classify those with visible fungal growth, those fungal strains as being pathogenic?
It's hard to say because just because they grow epiphytically is not to say we know what they're doing to the seed. Oftentimes the seeds germinate and are healthy.
Even with the like methods?
Yeah. In fact,
fact,
when I showed you initially the pictures of fungi growing from seeds, one of them was like this little yellow seed with pink. The pink indicates the seed is alive and the fungus is growing out of a living seed. So many times these seeds are alive and have fungi growing in them. In fact, I would say that's often the case. Any other questions? Yeah, Joe.
Because you saw distinct communities between leaf and seed in terms of the Endohyphal bacteria, do you think that those bacteria come along with that specific niche, or do you think that it's just a matter of influences?
Sure. So where do the bacteria come from? Are the communities different because the fungus is getting the bacteria from the leaf or from the seed? I would say that yes, and what I have been able to tease apart based on the bacterial taxonomy and information on GenBank where they came from, most of the bacteria, endohyphal bacteria among the endophytes, can be found living foliarly, or on leaves, sorry, is that word. And the other ones are often soil bacteria, so not necessarily associated with seeds per se, but they come from the environment the seeds are in.
Are there any other questions?
Eric?
So in the second chapter, your plant pathogen, you showed how it has the bacteria have very different effects on different substrates
Yeah.
for a single fungal species.
Right.
Do you have any idea of the other sides of the coin for other different fungi? How other fungi would be affected by bacteria?
I will say that the number of substrates that species of fungus was able to use is pretty high. Usually when you look at results of those plates, the number of red wells is not as high as that. So it has an extreme substrate diversity. So I would expect in other fungi, maybe the bacterium would, instead of influencing the level of which they use the substrate, they might determine which substrates can be used in addition to the similar patterns that we saw. But it's hard to say whether or not they would also influence or enhance substrates. Again, you'll probably see the other way.
Okay, so with that, I'd like to thank again all of you for coming and thank Justin for his presentation.